This is Joseph Frasca, and today my YouTube outline will be on Chapter 4, The Chemistry of Behavior, Segment 1. And I'm sorry about my voice, I'm a little congested today. So basically, we're going to start off with neurochemistry, the branch of neuroscience concerned with the fundamental chemical composition and processes of the nervous system. Neuropharmacology, the scientific field concerned with the discovery and the study of compounds that selectively affect the functioning of the nervous system. So now we're going to move on to receptors. They add a very important layer of complexity in neural signaling because any given transmitter may affect various kinds of receptors that differ from one another in structure. A receptor is a protein that binds and reacts to molecules of a neurotransmitter or a hormone. There are two different types: ionotropic receptor and a metatrophic receptor. The ionotropic receptor is a receptor protein that includes an ion channel that is opened when the receptor is bound to an agonist. A metatropic receptor is a receptor protein that does not contain an ion channel, but may, when activated, use a G protein system to alter the functioning of the postsynaptic cell. And there are also receptor subtypes, which are any type of receptor having functional characteristics that distinguish it from other types of receptors from the same neurotransmitter. That is very important. Any substance that binds to a receptor is termed a ligand and is one of the several kinds of effects. 1. A ligand is classified as an agonist initiates the normal effect of the transmitter on that receptor. Number 2. A receptor antagonist is a ligand that binds to a receptor that does not activate it, therefore blocking it from being activated by other ligands. And 3. An inverse agonist binds to the receptor and initiates an effect that is the reverse of the normal function of the receptor. Drugs with any of these three actions just described are properly called competitive ligands because they compete with endogenous transmitter for binding to the same part of the receptor complex. Non-competitive ligands may either activate the receptor, thereby acting as non-competitive agonists, or prevent the receptor from being activated by the transmitter, thus acting as a non-competitive antagonist. Some synaptic transmitters that are very important: amino acid. So a few examples of those are GABA, glutamate, glycine. Amines: acetylcholine, norepinephrine, epinephrine, dopamine, serotonin, melatonin. And gases: nitric oxide, carbon monoxide, which are soluble gases that diffuse between neurons to alter ongoing processes. At one time, it was thought that each neuron contained only one transmitter. We now know that some neurons contain more than one, a phenomenon known as neurotransmitter co-localization or co-release. The most abundant excitatory and inhibitory transmitters in the brain are amino acids. The most plentiful excitatory amino acids in the brain are glutamate. Glutamate is associated with excitotoxicity, a phenomenon in which neural injury, such as stroke or trauma, provokes an excessive release of glutamate that overexcited cells and eventually kills them. In contrast to glutamate, the amino acid transmitters GABA and glycine typically have an inhibitory effect. Acetylcholine was the first transmitter to be identified. Two families of acetylcholine receptors in the peripheral nervous system and the central nervous system. Nicotinic, referring to cholinergic receptors that respond to nicotine as well as acetylcholine. Most are ionotropic, responding rapidly and having an excitatory effect. Muscarinic, referring to cholinergic receptors that respond to chemical muscarine as well as acetylcholine. These are G protein coupled metatropic receptors, so they have slower responses and they can either have an excitatory or an inhibitory response. Move on to dopamine. Dopamine is important. There are 5 subtypes of dopamine in the body. The mesotriazole pathway, a set of dopaminergic axons arising from the midbrain and innervating the basal ganglia, including those from substantia nigra to the striatum. Norepinephrine, the two main clusters of neurons in the brainstem are the locus coeruleus in the pons and the lateral tegmental area of the midbrain. Norepinephrine-producing cells are said to be noradrenergic. Serotonin, also known as 5-HT. Only 200,000 of the 80 to 90 billion neurons in the human brain are serotonergic, but they exert widespread influence throughout the rest of the brain. Serotonin has been implicated in the control of sleep rate, sleep states, mood, sexual behavior, anxiety, and many other functions. Several families of drugs that are used as antidepressants share an action on serotonin. They act to increase its synaptic availability. For example, Prozac slows the clearance of serotonin from synapses by inhibiting its reuptake into axon terminals. And neurotransmitters as a gas. Neurons use certain gas molecules that dissolve in water, which are soluble gases, to communicate information. Nitric oxide is one of the biggest ones. It is produced in the cellular locations other than the axon terminals, especially the dendrites, and the molecules of nitric oxide are not held in or released from vesicles. The substance simply diffuses out of the neuron as soon as it's produced. It doesn't interact with the membrane-bound receptors on the surface of the target cell, but rather, it diffuses into a target cell and stimulates the production of the second messengers. And that completes my Chapter 4, Segment 1, YouTube outline.